Antibiotic resistance, as defined by the CDC, happens when germs like bacteria and fungi develop the ability to defeat the drugs designed to kill them. Well, how does that happen exactly? The answer lies in the natural process of evolution and natural selection on a bacterial level. To put it simply, a drug kills off every bacteria except those lucky enough to have acquired random mutations that allow them to survive the effects of that specific drug. These bacteria live on and reproduce, spreading their genes until the entire population is resistant to that drug. Well, what kind of mutations would allow a bacteria to survive a drug meant to kill them? This isn't completely understood yet, but we have a general understanding of the most common mechanisms. Most antibiotics target bacteria in several ways, such as stopping them from building cell walls and blocking metabolic pathways, among other things. But some bacteria may have a mutation or two that can find a way around these drugs. For example, they may be able to inhibit the enzymes that the drug uses to carry out these attacks. Or the bacteria could build alternate pathways to these important life cycle functions the drug was supposed to block. Once the bacteria can sidestep the mechanism of this drug, they are resistant. All they have to do next is multiply. The big problem that troubles the world's health experts is that more and more strains of bacteria have become more and more resistant to numerous antibiotics. If this trend continues, eventually there won't be any drugs able to treat bacterial infections. We would have a real superbug on our hands, and this problem isn't something that could happen far into the future. There already exist incredibly resistant strains all over the world. So, if antibiotic resistance is increasing over time, how do we stop it? Well, over the course of the past 80 years, we have been learning more about how to mitigate this. Before the early 1960s, pharmaceutical companies were easily capable of keeping up with resistant bacteria, either by creating new drugs or using drug cocktails. However, it was then realized that the continuous development of new drugs could not go on forever. Since then, antibiotic resistance has become a steadily increasing concern. This has led to a huge rise in research. For instance, to visualize the evolution of resistant bacteria, the Kishoni lab at Harvard University designed the megaplate. These researchers coated a gigantic petri dish in antibiotics with increasing concentrations towards the middle of the plate. Allowing the bacteria to grow from the outer edges inwards, they were able to spread towards higher and higher concentrations of antibiotics, eventually reaching the center with concentrations 1,000 times higher than normally survivable. This experiment was a great visual representation of how bacteria can continually become more resistant, and also a good lesson on why finishing a prescription is important. While we have learned quite a lot over the past 80 years, there is still a lot more to understand and a whole lot more to figure out if we want to solve the problem of antibiotic resistance altogether. We don't know what solutions we'll find, but with continued research and discovery, we can help keep these superbugs at bay.